Hello friends, welcome back to my channel in today's biology. Today we will discuss about the models of the plasma membrane. First of all, some introduction of the plasma membrane. Definition of the plasma membrane is the plasma membrane unclosed type of the cell, right? And composition of the plasma membrane is a protein 60 to 80% in the plasma membrane, lipid 20 to 40% in the plasma membrane, carbohydrate 2 to 10% in the plasma membrane, right? And the three properties of the plasma membrane. The plasma membrane is a selectively permeable variable protein and phospholipid ratio, and the plasma membrane is dynamic in nature, right? Two models of the plasma membrane. First, sandwich model, and second one is fluid mosaic model. First, sandwich model is proposed by Daniel and Davison. In this model, proteins are the alpha globular proteins and the phospholipid molecules are amphipathic, right? Amphipathic means one molecule is hydrophilic and second one is hydrophobic, right? And the phospholipid are arranged in bilayer in such as a manner tails face each other. You can see, right? In this model, the plasma membrane using the van der Waals force. You can see the figure. The head is glycerol head. Head is hydrophilic. Hydrophilic means love the water, right? And the two fatty acid tails is hydrophobic. It means hate the water, right? And you can see which was van der Waals force, which was helping the two layers stay together, right? In this model, one layer of protein and other layer of the protein and the between phospholipid bilayer, this based structure called the sandwich model, right? In this model, fake three lines. This model, the fat soluble substance, right? And the water soluble substance. Water soluble substance does not cross the plasma membrane because of the tails is hydrophobic and water is hydrophilic substance, right? So, and hydrophobic fatty acid tails because hate the water, right? So water soluble substance does not cross the plasma membrane in this model. The fat soluble substance cross the plasma membrane, right? In this model, fake three lines, this is not able to explain entry of the water soluble molecule, right? Second, this is not able to explain variable protein to phospholipid ratio. And third, this is not able to explain in the dynamic nature our plasma membrane, right? That's why this model was rejected, right? And the second model was fluid mosaic model, the proposed by the Singer and Nicholson using the information of the Daniel and Davison. And in this model also proteins are alpha globular, but alpha globular proteins classified in two proteins, extrinsic and the intrinsic, right? Extrinsic present on the outer side of the plasma membrane attached to the phospholipid head, right? And the intrinsic completely connected in the phospholipid bilayer, right? In this model also had glycerol fatty acid head is polar and fatty acid is non-polar, right? The phospholipid tails face each other and head are away from the each other, right? And in this model, the using the weak force attraction van der Waals force, right? You can see the figure. In this figure, the extrinsic protein and the intrinsic protein and the extrinsic protein. Arrangement is the protein layer is the protein layer, phospholipid layer, and the protein layer, right? And this model, in the water molecule substance can easily cross the membrane, and the fat molecules also cross the membrane, right? The fat soluble substance easily cross the membrane. Water soluble substance come by the intrinsic protein, you can see. And the two types of the movement in this model, transition movement and the flip-flop movement. So what is transition movement? The transition movement of phospholipid molecule is in the same layer, movement in the same layer, right? And the flip-flop movement, the movement in the between two layer, right? You can see, right? This is a transition movement. You can see 1 2 3 4 and the in the same layer movement, right? And the flip-flop means between two layer, right? In this model, allow the fat soluble substance and the water soluble substance, right? Second one is plasma membrane variable ratio. The phospholipid change the transition movement and the ratio has changed, right? Protein, in this model, the proteins are the proteins iceberg in the sea of lipid. That means comparing the proteins with the iceberg and the lipid with the sea, right? So all the information together is used by Singer and Nicholson. They call the models as the proteins iceberg, sea in the lipid, and this is accepted model. Thank you for watching my video. Please do like and subscribe to my channel.